In this discussion, we will talk about other signs of selection that we can find. Some of these do involve genes and locating genes, but we're not evaluating the content of the gene itself. So that's why I've put them under other signs of selection. Now, we've already talked about some signs of selection at the population level. So we talked about Tajima's D statistic, about F values like the FST, which looks for types of structure within a population. So those are also tests that look for natural selection, but on a little bit of a different level. And I think it's worth addressing what we mean by a different level and talking about that. So you will occasionally hear some people make a distinction between microevolution, which is defined as changes, variety, and evolution that occurs within a population. So here we have different breeds of sheep as an example. And then macroevolution, which are the changes and evolution that occurs between distinct species, or maybe we can say separate lineages. Now, this really is an artificial distinction. Just as the boundary for species can be difficult to define and really is impossible to draw a clear line. Now, that might come as a surprise to you, but if you think about it, it's really not a surprise because the vast majority of the time, speciation is a very long, slow process that occurs over tens of thousands of generations. As genetic differences build up between populations, and eventually we have enough of a difference between two populations to call them species. But it's not a clear, like, oh, boom, suddenly one day they are the same species and the next day they are different species. It's a long, drawn out process. So if that process has been going on for a very long time, it's not a hard distinction to make. So if I looked at an elephant versus an iguana, I'm not going to have any problems no matter which elephant I pick from the population and which iguana I pick from the population. I'm not going to have any problem drawing that distinction because it's been going on for so long. The speciation distinction is clear. However, if I were to pick, say, a forest elephant and a plains elephant, and in fact, that's a distinction that's been made fairly recently and was not recognized very long ago, I would have a much harder time drawing that distinction, and I might even have to resort to a genetic analysis of the two populations to determine whether they're truly different. And so, microevolution and macroevolution are sometimes a convenient box to put them in. Just like as humans, we will often say youth, right, versus adults, right? But recognize that the process of going from a child or a youth and becoming an adult is a long, drawn out process. And different cultures will mark different milestones along that pathway. But, just because you have a bar mitzvah or you have a manhood ceremony where you go out and kill a lion and then suddenly you're a man, that's an important cultural event. But even though it marks a distinction at the cultural level, it really is a long, drawn out process without any clear, distinctive changes, barriers along the way. Okay. Now, going from an adult to a child is a good analogy, but recognize there might be some times we can draw a distinctive barrier. But realize that what we call an adult is really an amalgamation of physical, cultural, and individual characteristics. So just because we draw a clear boundary in one, say, oh, look, the onset of menarche, which is the technical term for having your first period. So just because that occurs in a female and suddenly now they are able to bear children doesn't mean that emotionally or culturally they are an adult. And so the same way in species, we might be able to draw a clear distinction in some cases. That might be a good boundary in some areas. But maybe, for instance, if we say, oh, look, here are two species of plant that no longer can interbreed because there's a different pollinator, those artificial examples that we looked at draw a clear boundary between gene flow, but for all other intents and purposes, they are still very, very similar and might even be considered the same species if you weren't aware of that one single fact. So, in reality, there is not a clear boundary between microevolution and macroevolution. It gets very, very cloudy. And so if we can't draw a clear boundary, are these really clear distinctions? And probably the best answer is to recognize that, okay, it's useful in some cases, but really these are the same processes, perhaps at different levels, and maybe a little bit of different emphasis on some processes versus others. But really, evolution is evolution, no matter where it is occurring. Now, we have some good evidence of genetic convergence. And so this is one instance where we can see very clear patterns and trends, and that there's been clear pressure for very similar selection. One famous example is the example of the digestion of cellulose. It's a complex process, but one of the enzymes in mammals that allows for cellulose to be digested is a lysozyme. And this occurs in ruminants, including cows, and in a very small subset of primers that are able to digest and eat cellulose. And typically, this is a very lengthy process, usually involves the cooperation of other microorganisms, including changes in evolution in the genome of the cellulose digesters themselves. So it's a complicated thing, and often there are different communities of microorganisms, sometimes different genes, but in many cases we can see similarity in the genes that have allowed organisms to digest cellulose. So cows and other ruminants that can digest cellulose have a lysozyme that is part of the process allowing to do that. Now, all mammals have that lysosome, but it does not have the cellulose digesting function that it has in the ruminants, except in this small subset of primates. And it's very obvious all the other primates don't have cellulose digestion. There are many other mammals that are more closely related to the ruminants that don't have cellulose digestion. So it's very clear when you look at it at phylogeny that that ability to digest cellulose happened independently. And so when we evaluate the lysozyme that is involved in this process in these primates and in these ruminants, we find that there are 5 amino acid residues that mapped onto the phylogeny map at those independent lineages. And so those are convergent mutations. And not all of them are identical, especially if we look at the non-synonymous mutations that occur. We see very distinctive differences where the non-synonymous mutations of the primate lysozyme are more similar to other primate lysozymes, and the non-synonymous mutations of the ruminants are more similar to their close relatives. But the synonymous mutations that have been selected for are convergent and similar between these two. And then in the Hoatzin, which can also digest cellulose, it turns out that there is a paralog of this same lysozyme gene. So these two are in orthologs, but there's a paralog that has gained similar function, despite having a very divergent and different past. So it's a very cool example, and because we can now evaluate genes at the finest level, all the way down to the nucleic acid, we can recognize and find these very clear instances of genetic convergence. And as we look, we see many of these all over the place. So I'm just going to mention 2 others that we'll talk about in coming lectures. One is in opsins. Opsins are G-coupled proteins that allow for the transmission of a neurological signal after they have interacted with photons, with light. It's a fancy term for saying these are basically the proteins that give us our vision. And we see remarkable convergence in these it's a large family, larger in some groups than others. We see remarkable convergence in the wavelengths of light that they are able to detect. So different branches of this opsin family have independently gained the ability to detect UV light, for instance, in many groups. And then a third example is in the venoms of many organisms, particularly if they are targeting the same prey species. So they may have started with a similar complement of basic enzymes that can do a, maybe not really efficient, but an okay job of digesting or breaking down the tissues of, or interfering with other metabolic processes in the tissue of their prey. But then we see adaptations and selections towards more and more efficient of that. So opsins and venom are also good examples of convergence at the genetic level. So just like we see convergence in form and function at the morphological level, we can also see analogous things happening at the molecular level. There are a whole range of tests that are based on something called linkage disequilibrium. So let's define, hopefully it's a review, but if not, learn it. Linkage disequilibrium is the unexpected association of alleles or polymorphisms that are close to each other on the chromosome. So sometimes it's also called genetic linkage. And linkage disequilibrium is just where we find two physically close polymorphisms on a chromosome associated with one another more than perhaps we would expect based on their frequency in the population. And this is especially pronounced after a selective sweep. Now notice they've given a couple of varieties, a soft sweep, incomplete, complete sweep, and then a massive sweep. And so there are various levels of how this occurs. And there are two qualities to a selective sweep that are going to determine whether it's a soft sweep or a very hard complete sweep. And so let's list those two qualities and just then basically in short order we'll talk about why they would result in this difference between a soft and a complete sweep. So the first one is the advantage that a new mutation confers upon an organism. So at this point it's probably worthwhile redefining what we mean by the term selective sweep. When a new mutation occurs that is highly advantageous, it is going to be selected for, sometimes incredibly strongly. And again, it depends on the advantage that that one new allele confers on the organism. It depends on the genetics. Is it dominant? Is it recessive? Is it incomplete dominant? But depending on all those complicated factors, at times you get a massive advantage from a brand new allele. And when that occurs, it is swept to fixation in the population very, very rapidly. Now, recombination breaks up associations between alleles, but recombination, especially for very closely associated alleles, is going to be relatively rare. So if the advantage is very, very strong and recombination rates are relatively low, then we will get a very strong selective sweep. If the advantage is only minor, and if the proximity of an allele is not really that close, then we might have what we would call a soft sweep. Okay? But in this example, we've got these this advantage that they've indicated here with a 7-point star. And it rises in frequency very, very rapidly. And it takes these alleles that just by chance, right? So we've got little alleles marked in these red bars. And by chance, these two alleles were relatively close to the brand new adaptive trait. And so as it becomes fixed in the population, the alleles near it also become fixed. And we may even lose other alleles that maybe even conveyed an advantage, but the advantage of this new one outweighed the advantage of this one. But oftentimes these are polymorphisms or alleles that really don't have any clear, distinct natural selection advantage. And so what this means is we now have a short portion of the genome that is nearly uniform. Now, notice there have been secondary mutations, and again, these are often neutral, but secondary mutations that pop up. So we may get some differences, but we have a clear pattern in the alleles that were there originally with that new mutation and that are linked or swept along with it. Okay, so this is a selective sweep. It occurs because of genetic linkage, and thus we see this unusually high linkage, which we refer to as linkage disequilibrium. And so if we use a measure, this EHH is just basically a measure of how much diversity is near any one point along the genome. And so if this line is relatively flat, which is about 50-50, we would expect there's no linkage, no unusual association. But as natural selection sweeps the selected for allele along, we get an unusual spike in the level of genetic linkage measured. And so this spike means that there is some unusual amount of linkage in the population over that region of the chromosome. And so we know that something in there must have had a great advantage recently and be selected for. Now, this can also be extended in another type of a study that you may or may not be familiar with, but I'm going to spend a little bit of time reviewing it, again, hopefully for most of you. If not, you'll need to learn this. There are many others, and we have, because of our big data sets and ability to statistically analyze and more power from those statistical analyses as our data sets become larger and larger, there are lots of more complex tests that can be taken using linkage disequilibrium. I'm going to look at just one of these, a QTL analysis, but it's a very, very powerful one because it allows us to map and look for linkage disequilibrium across the entire genome. And the power of this is that we can use it for just about any genetically determined trait. So let's talk about the old way that they used to do it, and then we'll look at a new way, and a very powerful new way that's been used for the last decade or two. Originally, and this was mainly done with fruit flies, individual strains would be bred that were completely distinct and were essentially breeding true, much like Mendel bred his pea plants to breed true for certain traits. Fruit fly lineages were bred true for a variety of traits across genomes. Then, once we have these true breeding traits, we do a crossbreed. And then we would look at the offspring and we would map what parts of the chromosome they had from different parents, and then look for whether or not they would get the traits from one parent or another. And so we could then determine where there were certain parts. So if this fruit flies that have mostly red at this area have a particular trait in abundance, then that shows that that trait came from that part of the genome and vice versa. So we're basically using this crossbreeding approach, looking for the significant association of certain traits with regions of the genome. Now, obviously, we can't do this in humans for ethical and practical logistical reasons, right? We can't breed certain strains or races of humans and then have them crossbreed and look at their offspring. And so, but having large data sets allows us to get around this and not have the ethical problems. But we need a really, really big data set. So to do a QTL analysis, number one, you need lots of data. Number two, within that data, you need to identify genetic markers. These are typically, and not always, but typically are single nucleotide polymorphisms, commonly called SNPs. And we just look for areas, and they don't have to be specific for anything. It just has to be an area in the chromosome where some individuals, say, have a thymine, and other individuals have a guanine at that position. Or any two differences will work. Sometimes there's more than two. We might have 3 or even 4 differences at a single site. But if there's variation there, and if we have a large enough data set, we know about what to expect across the population. So if we have a single nucleotide polymorphism where 60% of the population has an A at, let's say, this site in the genome, and 40% has a T, then what we can do is we can look within subcategories of that population. And again, to be statistically significant, we need a pretty large sample. But so then we say, oh, what about people with predisposition for Alzheimer's disease? Do they have a 60-40 balance also? And most likely they do, because this site is not associated or linked with Alzheimer's disease. But if we have genetic markers that are spread across the entire genome, and now we've identified tens of thousands of them in the human genome, then we can look for that rare needle in a haystack where one of those genetic markers has an unusual level of association with a certain phenotype. So this is Alzheimer's disease. We have now identified 3 areas of the chromosome, and maybe a 4th with a little bit lower level of significance. But here's one that's very, very strongly associated. And so again, at this site, it might be like, a 50-50 distribution of this polymorphism in the population. But in the Alzheimer's population, 90% of individuals have a G, and only 10% of them have a C, where maybe it's 50-50 in the general population. And so we have identified a genetic marker associated with Alzheimer's disease. Now, the key to this is that polymorphism itself is probably not the thing that contributes to Alzheimer's disease. But because of linkage, we know that there's something nearby it. And the power of a QTL study is it could be an allelic variant, right, in the gene itself, or it could be a regulatory region near the gene. It could be even something else, right, maybe a microRNA that plays some role in it. But this does not have to be a gene itself. All we know is that somewhere in this neighborhood near this unusually associated marker, there is something that contributes to Alzheimer's disease. And so now we can narrow our focus down, look at the genes there, look at all of the differences that occur within the Alzheimer's population versus the population at large, and see a very clear, distinct difference. So recognize, to do this, not only do we need a large genetic database, but we also need a pretty well-defined medical history of individuals. And it can be anonymized, but we need to be able to know the genetic background, the medical background, family histories. And so we really need large data sets on both sides, both the genetic side and the medical background, or at least phenotype background of the individuals from which that genetic data has been gathered. And we're now getting those. There's a remarkable data set that's pretty widely available now from individuals in Iceland, where the Icelandic government funded basically DNA sequencing for a massive amount of the population, looking for these QTLs associated and anonymized so that people can't go in and identify who any one individual is by name. But you can use that. Now they actually are selling that to companies that are looking for genetic markers associated with a whole host of traits. And it can be really anything with a genetic background. So it's a very powerful tool and relies on linkage disequilibrium, right? So this genetic linkage between the markers that we have defined and the traits that are near them on the genome. Now, there are other really interesting patterns that have been discovered as we have gathered more and more data and been able to analyze it. So one of these is patterns of base composition across the genome and even within certain regions of the genome. So when I say patterns of base composition, what I mean is how often in the DNA do we see a GC pair versus an AT pair. And so sometimes this is also, it's probably, I guess, worth putting in here. This is also termed GC content bias. It's just another term for this bias that we sometimes see. Now realize from a neutral theory perspective, we would expect roughly 50-50, that there should not, over random mutations and natural selection working, we shouldn't, if there's no natural selection working on this differences in G's and C's versus A's and T's, we should not see a bias. It should be roughly 50%. So let's look at the data now that we have it. Bacteria have a fairly wide range in GC content. However, within a genome, it is largely consistent. So if I was looking at a bacterium that had a 25% GC content, right, fairly low GC content, I would expect to see roughly that same GC content over the entire genome. I would not have an area that was 80% GC and 75%. The entire genome would be 25%, except under rare exceptions. And we'll look at one little kind of wrinkle to this. So that means they have little or low intra within the genome, intragenomic variation. But between species, right, intergenomic variation, between genomes of different species, there's often very wide ranges of variation, okay? Vertebrates, on the other hand, have a reverse pattern. There's typically very little intragenomic variation. There's typically little very intergenomic variation is what I meant to say. So if I looked at the overall entire genome GC content of a cow versus a human versus a snake, I would expect to see probably not a very significant difference at all. But if I looked within the genome, notice they're hovering right around 50% here. But notice within the genome, there are these hot spots where we have a jump up in the GC content and then other hot spots where we have a jump down. And so when this is a significant over an area, we can mark it. And these areas that have a significantly different GC content are called isocores. So that's a good vocabulary word. So an isocore is the region of a genome that has a significantly higher or lower GC content than the rest of the genome. So overall, the genome is pretty much balancing out, but regionally we can see that. Now, when we see this, we know that there has to be some sort of natural selection because we have violated the rules of neutrality, which should put it right around 50-50. And so we can come back to the bacteria here and say, oh, there must be some sort of selection pressure on the entire genome of the bacteria to either raise in some species or lower in other species their GC content. So this is a really good sign for selection. And it turns out that there are various things that can cause this selection. But let me give you just one of the things that is an important factor, not in every case, but is often a deciding or maybe even the deciding factor in why we have a GC content bias. And this has to do with the chemical differences between a GC pair versus an AT pair. If you look at the DNA at the molecular level, hydrogen bonding is what allows nucleotide pairs on different strands to bind with one another. And the hydrogen bonds of a guanine line up with the hydrogen bonds of a cytosine, and the same thing for adenine and thymine. But with a GC pair, there are 3 hydrogen bonds that line up to allow them to match to one another. And in the AT pair, there are only 2 hydrogen bonds. So we have more stability between GC pairs, right? Thermodynamically, chemically, they're a little bit more stable. Hydrogen bonds are weak, but it's a significant difference of 3 versus 2. And so more thermodynamic stability in GCs mean that if we are living in an environment or if there is a condition for more stability of that region of the DNA, like in a genome, then it will, over time, be elevated as random mutation brings in G's and C's versus A's and T's. And so we would see an elevation. So often these elevated regions, these isocores in genomes, are selection for more or less thermodynamic stability. And in bacteria, we'll often see an association with the habitat that they live in. So organisms that live in a very extreme environment where DNA needs to be very thermodynamically stable, maybe a hot spring or a high salt concentration. This occurs in many archaea also. There would be a selection over time for higher and higher GC content. If, however, you're living in an area where we need less stable DNA, we need to be able to open up and access genes or replicate DNA more easily in those environments, then we maybe a very cold environment. We would need a lower GC content, and over time it's been selected for, and that increases the efficiency of any process that needs to unzip the DNA. Okay, so we've identified these isocores in different vertebrates, other eukaryotes also, and they are clear and distinct. So in the human genome, we see, these distinct areas that are GC rich, and these other distinct areas that are very GC poor. Okay, and again, some of these have distinct reasons why, others we're not sure, but there's a clear sign that something is going on, and then the question becomes, how do we identify what the process is that has led to that? Okay, and so here's just an example of some of these GC rich versus AT rich areas that occur in different organisms. I believe this is one that was primarily identified from Drosophila. Actually, this might be human. I'd have to look it up. But the idea is that there are multiple different things that might lead to a GC rich versus a GC poor area. Okay, and so all of these selective pressures, sometimes more than one acting on a single site, lead to these isocores in vertebrates. Now, so it is listed right here. So this is vertebrates looking at humans and maybe other organisms. And we might see other areas that are present in, arthropods or other invertebrates. So it's not something unique to vertebrates, but they can be identified. Now, I don't expect you to memorize any one of these, but I do want you to know that there are various forces that can cause it, and maybe know the stability of the DNA may play a role, where if there's selection for more stable DNA, we would tend to see GC-rich region, or maybe a GC-rich genome in the instance of a bacterium or archaean. And that if we need less stability, then we may see a GC-poor region. Okay, and so these isocores can be based on mutation rates and if there's bias in DNA repair. So it might not be really any sort of natural selection at all. It just might be, there's some sort of a bias in the mechanisms that are generating the patterns. And so that's something that we cannot always rule out. But many of these we have now identified as selectional cause for them, either thermal stability or gene expression area that needs to be unwound very, very rapidly and commonly. And so many of them do have selectionist ones. There are some that we have yet to define or find a selection reason for why that isocore exists. And so in that case, it may be, and we may need to then start consider, maybe there's a bias in the processes that are repairing the DNA. Or maybe there is some bias in mutation, although generally we tend to think of mutation, and in most cases it clearly is a random mistake that happens with no bias towards one base pair or another. The last pattern that we're going to look at, selection pattern, is called codon usage or codon usage bias. And again, from a neutral theory, which is our null hypothesis, if there is more than one codon possible for a amino acid, we would expect to find it across the genome in roughly the same frequency. And this can be tricky because of data set size. If we're just looking at one gene, there may not be enough threonines, for instance, in that gene to say whether it's biased. So if you have only 7 threonines in a gene and 5 of them are one codon, and I think with threonine, with threonine there are 4 possible ones. So if you see 5 out of 7 threonines are ACA, that's probably fairly significant, but it's a large margin of error when our sample size is only 7 residues in the amino acid. So most of these studies are done over many, many genes, not just a single gene. But basically what we're looking at is some bias. So threonine, which has 4 amino acids, we would expect each amino acid to be roughly there at 25%. right? So we would expect to see this 25% level. I'm not sure actually what, maybe that's 50% of humans versus E. coli. Yeah, so notice here in the leucine, there's a clear bias for this CUG, and it's actually stronger in E. coli than it is in humans, but there's still this clear bias where we see an unusual amount. Instead of them being all about the same, this one, the CUG version of leucine, tends to be at a much higher level. Okay, and so then the question arises is when we see a bias, why? Why are some codons represented at higher rates? And what we typically find is that this is often associated with increased expression of the genes in which we see a bias. So if we broke this down, maybe the genes that are highly expressed have even more pronounced bias, or maybe they have a bias and the low expressed genes don't have a bias. And so this association between codon usage bias and highly expressed genes was the first clue that allowed us to identify what was going on. And it actually correlates, it's not the exact same thing, but correlates with something that students are often more commonly aware of, which is the wobble hypothesis. And the wobble hypothesis also helps to contribute to translation efficiency. So we're looking for an increase in the efficiency of translation process. I might have said transcription earlier, but it's actually the translation that we're looking at here. And so the idea is that if you have 6 different codons, anytime you need a leucine, it could be one of those 6, and that there is a specific tRNA associated with each one of those 6 codons, that's going to, in maybe a fractional but still a significant amount, is going to slow down the process of translation of this mRNA into a final protein product. Just as if you were building a Lego structure, a sculpture, let's say a character out of Legos, and if any time you needed a particular type of brick, there were 6 different containers that you could draw from, that's going to maybe make a little bit of a pause as you're looking for those 6. Whereas if you know every single time you can always go to the same one, or almost every single time go to the same one, it's going to increase your efficiency of completing that process. And so with translation, it's the same thing. So most of the time, nearly always when we see codon usage bias, it increases translational efficiency. And this occurs across many, many other organisms. So highly expressed genes need more translational efficiency because they're just going like crazy. So they use fewer tRNAs, and then there is a codon biases. So both the tRNAs that are present and the codons that are present are biased together to mean that we have a more efficient translation of the nucleotide. And so this means then there might also then be pressure on what we would normally think would be a synonymous mutation, right? So now suddenly that third position, which you would say wouldn't make a difference because we'd still have leucine, now suddenly it makes a difference. And so this codon usage bias is not only in the coding differences. That's obviously can be a clear difference. If we mutate this uracil in the middle, this is at the RNA level, we're going to get a different gene. But now if we mutate the third one, we end up still with leucine, but a difference in translational efficiency. So this also goes hand in hand with wobble hypothesis, although it's not quite the same thing. Wobble hypothesis just basically means that the ribosome is not completely high fidelity for one type of tRNA, that at the third position, the ribosome will often accept a tRNA that also has it. So we never completely eliminate all of the others, and they can slide in with not 100%. So for instance, if the tRNA is CCC and it slides in very easily and matches to that RNA complement, which would be GGG in the RNA, right? Now we get a GGC that comes in, and still the CCC might match with it and fit well enough that it works. And so that's the wobble hypothesis. And so both wobble and codon usage bias go hand in hand to increase translational efficiency, and it is especially pronounced with this codon usage bias in these highly expressed genes.